Hi, Brian Koffman, a retired family doctor and a CLL patient myself, and the Chief Medical Officer at the CLL Society here on the last day of ASH 2019 in Orlando, Florida.
Hey Brian, I'm Krish Patel. I'm the Director of the Lymphoma Program at the Swedish Cancer Institute in Seattle, Washington.
Dr. Patel, there's been a lot of excitement, not just in CLL, but in lymphomas in general, in fact in cancer in general, about harnessing the immune system. So before we get to specifics, can you tell me why, as a cancer researcher, you're excited about using the patient's own immune system?
Yeah, absolutely. I mean, I think we see that there are really powerful ways that the body itself has the ability to eradicate cancer cells, and in many patients who have cancers including CLLs and lymphomas, the immune system becomes dysregulated in a way where it's not able to do that. So we're really just starting to find the right tools that allow us to really unleash the native immune system's ability to eradicate the cancer. So I think it's a very exciting way to be able to treat it.
And there's all different ways this is being done. There's drugs that take the brakes off the immune system, checkpoint inhibitors. There's drugs that modulate the immune system. There's cellular therapies which have used the body's cells that have been sometimes genetically modified, or sometimes cells from someone else who attacked the cancer in a transplant kind of case. But the antibody area is very interesting. And there's a new kind of antibody, a new kid on the block called a bispecific antibody. So these are similar but different than the things we're used to, like rituximab and obinutuzumab. So first, remind us what a monoclonal antibody is and what the difference between that is and a bispecific antibody.
Absolutely. So a monoclonal antibody, which rituximab is really the one probably most are familiar with, really has one antigen that it's binding to. It's binding to an antigen on...
and an antigen is a protein on a cell.
On a cancer cell. It's binding to a protein on a cancer cell, and then it's kind of hoping that the rest of the immune system will recognize that it's binding that cancer cell.
Waving, I'm here.
I'm here, I'm here, destroy this cell. A bispecific antibody takes that, I think, to the next level, where one side of the antibody is binding to that protein that is on the tumor. The other side of the antibody is binding to a protein that's on the immune system. So it's like saying, it's like handcuffing the immune cell to the cancer cell and say, hey, come here and I want you to take care of this problem.
And is this overly simplistic, but as I'm remembering from immunology, that these antibodies are often Y-shaped, so is like one part of the Y attacking the cancer and the other is attaching to the immune system?
That's right, and now we're able to do very cool things with antibody engineering. So the basic premise is exactly that, but you can even actually have even more complex structures beyond the traditional Y that might allow you to better bring the immune cell in contact with the cancer cell.
So without getting too technical, what are the targets that you're using for the cancer and for the immune system on this double antibody sort of?
So in CLL and lymphoma, we're using CD20, which is the antibody that rituximab recognizes, so it's a well-known target. And then we're using CD3, which is a protein found on all T cells. So we're bringing T cells to the cancerous B cells.
And tell me where this is at in trial and what results you have.
So this was an exciting ASH, I think, for bispecific antibodies. There were several presentations, and there's a couple different compounds that are out there, all of the CD10 and CD3 variety. We've been...
So CD10 would...
Or sorry,
sorry, is that right?
CD20.
CD20, okay, all right.
And CD3.
Okay.
And there's we've been working on one in particular that is being studied in patients with CLL as well as other B-cell non-Hodgkin's lymphomas. And what we're seeing across this class of drugs is that they're highly active in patients who have had lots of other therapies, including some patients that have been previously treated with CAR T cells, so that's exciting.
Wow.
And in general, I would say that they have a side effect profile that is fairly consistent. And those side effects are mostly immune-stimulating side effects like cytokine release syndrome, which is something we've seen and experienced in other immune treatments like CAR T-cell therapies.
Wow, so you get these cytokine release syndromes. Do you get, I have to ask, do you get any of the neurological events that you...
So it's interesting. What we see is a little bit different with bispecifics. So we don't see the kinds of neurologic side effects in as frequency with bispecifics, so it's much less common for us to see those neurologic side effects with bispecifics. And the severity of the cytokine release syndrome also seems to be lesser.
So I know the numbers are very small, but do you have any results that you can share with us?
Yeah, so the agent we've been working on, it's a bispecific antibody by Xencor. We presented safety data for about 55 patients or so, and we presented efficacy data for patients with diffuse large B-cell lymphoma. What we saw in safety was that cytokine release syndrome was generally mild, mostly grade 1 and 2, and the agent was very well tolerated across different patient groups. In terms of efficacy, what we see, it's still undergoing dose escalation, but at higher doses, we're seeing very encouraging activity in diffuse large B-cell lymphoma, where people are actually having complete responses to treatment, and some of those patients are maintaining those responses over time. So we'll see how that goes in the long run, and we're continuing the dose escalation.
So diffuse large B-cell lymphoma tends to be a more aggressive cancer than CLL, but you have trials in CLL.
We do, and so we didn't report efficacy data on the CLL population because it's small in the study. We had 8 patients, but one thing I can say that was very exciting and included in our poster is we had a patient with Richter's transformation, which can be an aggressive transformation of CLL and quite difficult to treat, and that patient also had a complete response, which we were quite optimistic about.
Yeah, that's amazing, because Richter's remains one of the great unmet needs for
absolutely.
CLL patients with a generally dismal prognosis for people who get routine options. Now, so is this trial open now?
It is. So the trial is continuing to enroll patients both with CLL and non-Hodgkin's lymphoma. In the CLL study, we're actually a little bit behind in the dose escalation, so we're continuing to try to find the right dose in CLL. It may be different than in HL.
Okay.
But the study is actively enrolling, and we look forward to learning more.
And Richter's, can patients come
That's
in
correct.
with Richter's too?
Yeah.
So make sure I have the information for patients through clinicaltrials.gov, or
Absolutely.
if there's a direct website. And is this a multi-center trial or just at your site?
This is a multi-center trial, so there are sites in the U.S. as well as in Europe.
All right, good. Very exciting. Any final thoughts on this bispecific antibodies and where things are going?
Yeah, I'm very excited about this space. We recognize that some of our other immune treatments can be a little challenging to deliver, like CAR T cells. And I think that because bispecifics are antibodies, a type of drug that most practitioners are familiar with, they have the potential to really be used in more patients, hopefully closer to their own home communities. So I think it's an exciting space to watch.
I'm very excited about this research. Thanks so much for what you're sharing.
Thank you,
Thanks.
Brian.
Yeah.